So that's all the stuff that you can see pretty much with the unaided eye. Now we dive into the microscopic anatomy. A lot of things here you've seen. Cytoplasm, plasma membrane, T-tubules, sarcoplasmic, smooth ER. Okay, a lot of the stuff you've seen, it's just unique for skeletal muscle, okay, or unique for muscle. But we're talking about skeletal muscle, so let's stay there. So we have a muscle cell, and again, muscle cell, muscle fiber. I think this is one of the sections where the book interchanges them. Okay, but we have cytoplasm found only in a muscle cell, and it's referred to as sarcoplasm. Sarc means flesh. If you've ever eaten meat, there you go. That's where the name came from, like in ancient Greece. Sarcoplasm. It's cytoplasm. Okay, has our typical menagerie of organelles, inclusion bodies, okay, but it also has a lot of other specialized, contractile proteins. Obviously, it's contracting, for God's sakes. We have a plasma membrane of a muscle cell, muscle fiber, and we refer to that as a sarcolemma. Okay? So when you're talking about it, you can't say plasma membrane of a muscle cell or plasma membrane of a muscle fiber. Got to call it sarcolemma. Right? That is the plasma membrane of a skeletal muscle. You will find some unique things along the surface of that sarcolemma. One of those is an opening called T-tubules. I have it shorthanded. If you want to spell it out once on the exam, if I'm asking a short answer question on skeletal muscle contraction, right? And you want to talk about the T-tubules, yes, you can. Spell it out. And then if you want to do shorthand with T-tubules, yes. But I need to know that you know what that T means. So please spell it out once and then use shorthand. These transverse tubules open up on the surface of that plasma membrane, and they run down deep into the muscle fiber. It's going to be vitally important, this sarcolemma, these T-tubules, okay, for getting electricity down into the muscle fiber, into that sarcoplasm, to allow magic to happen there. We have a number of voltage-gated ion channels along it as well. And I'll just write them here: sodium and potassium. Okay. We have a number of sodium and potassium voltage-gated channels. They are going to be vital for how electricity travels across the surface of this muscle fiber. Another thing that we have here as well are calcium voltage-gated channels. These are three huge parts associated with how muscle contraction occurs. So what I'm saying is you should know these things because you're going to be talking about them quite a bit. And just as a side note real quick, we'll have the nervous system next. Nervous system, neurons have their own plasma membrane. Neurolemma. Whoa, mind-bending, right? But it's going to have the same stuff. It's going to have voltage-gated channels on it. Sodium, potassium, calcium. So this is stuff that's going to be very common that we see in this unit, in the next unit, in the unit after that with cardiovascular, in the next unit after that with the digestive system. So these are things, guys, gals, that we're going to be talking about a lot from here on out. Understand this now, and all you have to do is do a slight little tweak when it comes to cardiac muscle, a slight little tweak when it comes to smooth muscle, a slight little tweak when it comes to the nervous system. Okay? But it's the same thing, same concept. We just start adding and taking away a few things. All right. So learn it now, make it a hell of a lot easier later in the semester. Please, trust me. So this is what this looks like. Phospholipid bilayer. Yep, sarcolemma. And then here you can see we have voltage-gated channels. There's this right here with my calcium voltage-gated channel right here. Okay, there's my calcium. I'll just do voltage gated channel. Here. Oh my gosh, excuse me. There you can see the sarcolemma has been reflected away, so you can see some of the fun stuff. And here is that T tubule. There's just that opening along the sarcolemma. Okay. And this is the sarcolemma, it's the plasma membrane. On top of this is the endomysium. So it further helps to insulate this thing. And here you can see down here, they kind of keep teasing it out to see all the little fundamental pieces. We'll be doing going into all this here. So wrapping around a skeletal muscle fiber is the sarcolemma. Within the sarcolemma is sarcoplasm. On the surface of that sarcolemma, I have a lot of things, 3 things: voltage gated channels, sodium, potassium, and calcium. All 3 going to be involved in this process called muscle contraction. This whole thing right here is a muscle fiber. Okay. See, there's the fascicle, and fascicle is clumping a bunch of muscle fibers together. Each of this muscle fiber, as you can see, is made up of myofibrils. And as you can see down here, the myofibrils are comprised of even smaller stuff called myofilaments. The entire muscle belly, bundled up, subdivided into fascicles, which is just a group of muscle fibers. When I look inside a fascicle, I can pull out that individual muscle fiber. It has sarcolemma around it. It's comprised of many myofibrils. And myofibrils are comprised of even smaller stuff called myofilaments. And we'll be talking about really all of these things when we talk about muscle contraction. Myofibril is just a collection of myofilaments. As you can kind of see, they're enclosed. There's hundreds of thousands of them per cell. I'm not going to ask the number. It's just there to kind of say, holy smokes. But these are all my myofibrils. And if I go back here, you can see all of these myofibrils are made up of smaller things called myofilaments. And it's these myofilaments that are wrapped in this sarcoplasmic reticulum. Let me go back here. Okay. Here is that sarcoplasmic reticulum. And as you can see, it's completely wrapping those myofilaments. It completely wraps around a myofibril bundle. There you go. As you can see, myofibril just means bundle of myofilaments. I don't know why they just don't call it that. You've seen sarcoplasmic reticulum before. It's basically smooth AR that's specialized for skeletal muscle. It's specialized because it holds calcium ions and it doesn't do anything involved with making fats. At the end of a sarcoplasmic reticulum, so let me go back here. So here you can see all this kind of purple stuff right here. This is the sarcoplasmic reticulum right there. You can see towards the end, near each T tubule, we get an expanded end of that sarcoplasmic reticulum. That expanded end of the sarcoplasmic reticulum is the cisternae. And it's at the end, so they give the moniker terminal cisternae. Okay, sure. It's just the expanded end of a sarcoplasmic reticulum. That's all. Both of these dudes The sarcoplasmic reticulum and the terminal cisternae do one thing. Again, store calcium. That's all they do. Sometimes they refer to it as a triad, the two terminal cisternae, terminal cisternae, terminal cisternae, sandwiched between the T tubule. I guess, sure, okay, yeah. I mean, am I going to really ask you to talk about that on a short answer question? No, I got a hell of a lot more things I can ask you about. Okay, but this is a triad. Other than that, I'm not going to, get into it a whole heck of a lot more. All right, but that's what a triad is. The two terminal ends of the cisternae that wrap around the T tubule. That's it. Which is great. I mean, it is good. I'm blowing it off as it's not good, but I mean, electricity travels down this thing. As electricity travels down this thing, okay, it can open up two terminal cisternaes. So it can go down one end and open two. Instead of having a huge space, having a cisternae way over here, I'd have to have another T tubule here, and it wouldn't be called a triad at that point. I wouldn't be talking about it. But this triad is, here's the T tubule, and it's how electricity travels down. And as it travels down, it tickles this guy and it opens up. And as you can see, it releases calcium. Oh, there's some stuff in there. Calcium enters into the cytoplasm through those voltage-gated calcium channels. And there's a couple enzymatic actions through calmodulin and calsequestrin. Calmodulin and calsequestrin are really there to kind of help exert the ability to hold on to calcium inside it effectively. And inside it, I mean inside the terminal cisternae. And it's also going to be involved in getting that calcium to come back home. Because again, when a muscle contracts, I need it to relax. And calmodulin and calsequestrin are going to be helpful in that process. Smooth muscle has calmodulin a little bit more involved with it. So it's stuff that we'll talk about again and mention briefly. Here in skeletal muscle, I don't get into it as much as I will with calmodulin with smooth muscle, but it is two things that exist within the cisternae that help to bring back in and help to hold on to calcium within it. It just doesn't go willy-nilly on its own. Okay, it has to be recalled because of these two things. It has to be held on to because of one of these things. So calmodulin, calsequestrin are things that we'll discuss. We'll discuss it a little bit more in terms of smooth muscle, though. So as we keep kind of descending into the abyss here, we have the myofibril wrapped around with sarcoplasmic reticulum, terminal cisternae, all that fantastic stuff. Myofibrils were just a bunch of myofilaments. Finally, we've gotten to the end. Myofilaments is where the magic's happening. These are contractile proteins. And we have ultimately two types that we'll talk about: actin and myosin. There are other ones, and I'll mention them here as well, and they'll be involved a little bit. There's another one. There's a few other. There's actually like 3 or 4 of them. 1, 2, 3, 4, 5. There's like 5 different thin filaments that we have inside skeletal muscle. We're not getting into all of them and what they do. We're just looking at some of the major players here. So we have myofilaments that are really big, ingeniously referred to as thick filaments. This is myosin. And there it is. There she blows. Big old thick. See, it's got a bunch of globular heads coming off of it. Ooh, a whole bunch of fun stuff there. Thin filaments are much smaller. Thinner. I didn't want to be that guy, but I'm going to be a guy. Thinner. Actin. And as you can see, this actin molecule is these two kind of helical monomer chains stuck together. They're made up of a bunch of a G-actin protein. Okay. I'll just do this. G-actin, F-actin, I don't care. Okay. They are a bunch of actin monomers strung together in a helical shape. That's all it is. And unless you're doing upper level work, getting into G-actin and F-actin isn't really necessary for us at this point. There are two other ones on here. Aside from actin, we have troponin and tropomyosin. These two are regulatory proteins. You only find them on actin. They're only associated with the thin filaments. But there you can see tropomyosin, like a rope. Troponin is like little bedazzled beads along the rope. Okay. But these guys, these troponin and tropomyosin, are important because, as you can kind of see from this picture right here, calcium is going to bind to troponin. Magic's going to happen. As you can kind of see, that green rope is over these little holes here on those G-actin molecules. Those are myosin binding sites. So as we'll come to find out here, when calcium binds, that causes troponin to change shape, which causes tropomyosin to move. And I expose those myosin binding sites on the actin molecule. Who do you think goes up and binds? That homeboy right there, that myosin globular head. So in terms of muscle contraction, I needed to travel across a sarcolemma, electricity. I needed to travel down the T-tubule. I needed to open specific voltage-gated channels to allow calcium to be released from sarcoplasmic reticulum and terminal cisternae. I need that calcium to be entering into the sarcoplasm. As it does, it's going to interact with troponin, moving tropomyosin, allowing actin now and myosin to interact to perform what's called the power stroke. Fancy. And all that means is contraction. That's it. But it's all these little parts and pieces. If you know what all these little parts and pieces do, the muscle contraction sequence is actually a pretty easy question. But if you don't know what myofilaments are, they are the contractile proteins. If you don't know, there's thick ones called myosin with globular heads, thin ones called actin with myosin binding sites on it, with troponin, with tropomyosin on it, and you don't know what they do, then I guess the muscle contraction sequence is hard. And for some students, it is. So there's our muscle fiber. And as you can see, I've teased out a bundle of myofilaments, myofibril. As we strip away sarcoplasmic reticulum terminal cisternae, you can see there is a wonderful repeating pattern here. If it's a repeating pattern, then you darn well know that we can measure this now. We can see the process actually happening. And depending upon how good your microscope is, you can actually see it. I took a phys class in my undergrad, and you could make stainings to this. You can introduce electricity to a muscle fiber on an animal, and you can do a different technique to stain this and see how all of these components move as you introduce electricity and the muscle contracts. Because this is in a nice, fantastic repeating pattern, we organize this thing into what's called a sarcomere. So really, when you look at those myofibril units, those myofilaments, they're actually organized into this sarcomere. And as you can see, it gives it the very characteristic look to it of that striped appearance, both this and cardiac muscle. They have the same arrangement. Now, there's some few changes from some of the anatomical structures that we just talked about, but they're still in this repeating unit of thick and thin, thick and thin, thick and thin, to give it that striped appearance. What defines the beginning and end are Z-disc. This defines a sarcomere. From one Z-disc to the next is a sarcomere. And this is where all the functional parts of the skeletal muscle are found within a sarcomere. Where these things are important. Not necessarily so much here because a muscle contracts and then it relaxes. And as we go over some of the major parts here, it's going to be very obvious kind of what they do and what's there. But when we start looking at the heart, it changes dramatically some of these parts within the sarcomere, which is important for how the heart muscle does what it does. And does what it does even as you put more stress and strain on it, which doesn't make any hell of a sense now. But it will when we start talking about this, and then we start looking at the cardiac muscle here at a couple units from now. Because we'll be talking about all these things again in the heart. I know, God, right. So these are our major kind of parts associated with the sarcomere. I bands, A bands, H zone, M line. There's some other lines that are kind of associated in there as well. But these are the major ones. What I have underlined, and I don't have this one underlined. I usually like to ask what defines a sarcomere. Z line is, or Z disc to Z disc. Okay. I like to ask, what's found there? Thin, thick, both. I like to ask a few of those things. So an I band is only thin. Okay. Let's go here. Okay. And here is my I band. There it is right there. Okay. Right there. There's my I band. Right there. Right there. As you can kind of see, all of this, and this is also thin filaments as well. Okay. It's just referred to as connectin. That's all it is. Right. It's just another smooth muscle. Connectin is, as you can kind of see, it attaches the myosin fiber to the Z disc. So it helps to hold that myosin fiber in place, or myosin filament in place. But that is my I band. It is all thin filaments. Okay. It will actually get, do I have a picture? Oh man, I took a picture out. It actually gets smaller when the muscle contracts. Okay. I mean, as we contract, these two things are interacting with one another. So this is going to get, this will get smaller as the muscle contracts. And that means this Z disc is getting closer, So this myosin, this coil is contracting and pulling that myosin over. A band, let me go back here. There's the A band. As you can see from the picture here, we have the M line right in the middle of it, and this basically bisects the sarcomere, divides it in half. No big deal. But you can see we have both thin and thick filaments here. Contains both thin and thick. I'm going to color one part in here real quick just to kind of highlight it. Also within this layer, we will have a what's called a zone of overlap. And I'll just do this. Zone of overlap. So here we have a zone of overlap. And as this muscle contracts, that zone of overlap is going to get bigger and bigger and bigger. And that H zone is going to get smaller as this thing gets scrunched in together. Again, the H zone is only thick filaments. Again, you can see here, only thick. Right? No actin there at all. And as the muscle maximally contracts, this will get a heck of a lot smaller. Damn near disappears with pure maximal contraction, which semantics on if that truly exists in normal anatomy. The last thing is an M line. And there it is right there. As you can kind of see, it just bisects it. It just, we have the connectin to anchor it down on both ends. The M line helps to hold it in position medially. I mean, that's it. It doesn't really do anything. When the muscle contracts, as the sarcomere gets smaller, I mean, it stays there. It doesn't really do anything. So, I mean, I don't really get into it. What defines a sarcomere? What defines these very structural units of this functional thing? The Z disc. In the Z disc, we have an I band. This is thin filaments only. It disappears as the sarcomere contracts. We have an H zone, right smack dab in the middle. Gets smaller as the sarcomere contracts. Have the A band with its zone of overlap, which actually does enlarge when the muscle contracts. And you can see the A band has both thick and thin filaments associated with it. But because it is a repeating thing, it's something that can be easily tested, easily studied to figure out what in the world all this does. Here's my other small muscle proteins, myofilaments. Okay, these are thin. Connectin, like I said, just helps to hold that myosin myofilament in place. Dystrophin is the other one. It's like the glue that holds it all together. When you have Duchenne's muscular dystrophy, you don't produce dystrophin, or you produce a little bit of it, and then you gradually, as the muscles contract, you don't make any more. And then the muscle can't work anymore because it's not glued together properly. So it all frays apart. So with Duchenne's muscular dystrophy, that's just one type of muscular dystrophy. And with that one, incurable. I don't believe there's any stem cell. I don't know if there's any stem cell research on it or not off the top of my head. But people who have Duchenne's muscular dystrophy usually don't live past like 30 very much, just because of the massive breakdown. I mean, it breaks down the diaphragm too. It's a muscle. That's a muscle that helps you breathe. and you got to breathe. The last little piece of microscopic anatomy is really more just kind of the fine tuning of it. They generally have a lot of mitochondria, some more so than others. If we have a lot of mitochondria, that means, well, we have a pretty extensive blood vessel network there, and we already established that earlier. What's all this allow us to really do? I mean, to use oxygen effectively to make ATP. If we have a good blood supply and we have a lot of mitochondria, that means we're going to have a lot of oxygen inside these things, or more oxygen inside these muscle cells than really any other cell. So we have this stuff called myoglobin. Myoglobin is what holds on to oxygen in a muscle fiber. Myo means muscle. Globulin is just the protein that holds oxygen. So booyah, there we go. Hemoglobin does it in red blood cells. It's the same thing, just about. Okay. We have glycogen. Glycogen is our storage form of glucose. Glycogen is our storage form of glucose. Skeletal muscles can store some glycogen. Obviously, they're contracting. Glycogen is glucose storage, again. So when your muscles need energy, carbohydrates, glucose is that primary fuel. They pull it out of storage. Okay, they just clip a glucose molecule off. Glycogen is just thousands of them strung together. Clip off a little glucose molecule, boom. Easy, quick energy source for the skeletal muscle to make ATP. The liver also is another huge store. Those are our two biggest stores. Once we fill those up, where does all that excess sugar go to? The F word. And nobody likes the F word. Fat. Another thing that skeletal muscles have that we'll talk about a little bit later is this thing called creatine phosphate. Phosphocreatine could be written down like that in books as well. They flip-flop. Creatine phosphate, phosphocreatine. It's the same thing. Creatine phosphate we find a lot within skeletal muscles. Good stuff. It's a phosphorylator. Creatine has a phosphate attached to it. That's it. We take that phosphate from creatine and smack it on an ADP, and we've made ATP in the process. Very quick ATP supplier. How quick? Like 5 to 10 seconds. That's really fast. So skeletal muscles have a great deal of adaptations that we see in them because of what they do. They contract, some quite a bit, some not as much, some with a lot of force, some with not as much force. All of these things require many adaptations, a lot of mitochondria compared to a typical cell, something to hold on to the oxygen, myoglobin, store for quick energy, glycogen, and a way to make fast, dirty, easy, quick ATP, creatine phosphate. So these are all adaptations that we'll see here within skeletal muscle that will allow it to do what it do. Is this the same for cardiac muscle and for smooth muscle? It's a little different. Okay? It's a little different. And we'll get into those differences maybe at the end of this lecture, but definitely when we get into those two units.